Hello everyone, my name is Hung Liang Pai, a medical student from Chung Shan Medical University in Taiwan. Today it is my honor to be here to share my research with you. The topic is incidence of ocular diseases as related to hyperuricemia and gout based on Taiwan Biobank. Oxidative stress resulting from reactive oxygen species, or ROS, has been implicated in the pathogenesis of various ocular diseases. Although emerging research provides some evidence of the important physiological role of ROS in normal cellular function, disease may occur when ROS concentrations exceed and overwhelm the body's natural defenses against them. Previous studies have revealed that overexpression of ROS can lead to chronic inflammation, which is implicated to induce ocular diseases, especially related to aging. Therefore, it is reasonable to deduce that other conditions related to inflammation and oxidative stress, such as hyperlipidemia, cardiovascular disease, hyperuricemia, and cancer, can increase the risk of eye abnormalities. Recently, hyperuricemia has been hypothesized to be a factor of age-related macular degeneration. However, no sufficient evidence in single nucleotide polymorphism variants is provided to its association. In this study, we investigated this potential relevance based on Taiwan Biobank. The Taiwan Biobank is a large-scale national biobank that supplies valuable phenotypic and genetic information to biomedical researchers among Taiwanese population. This community-based database has obtained International Organization for Standardization, or abbreviated ISO 27001 and 29100 certifications for governance and personal data protection, providing guarantees for biomedical research. In this study, the participants who completed questionnaire, physical examination data, blood and urine test data, whole genome typing data were enrolled. Individuals who met the following criteria were excluded. Those who were originally blind, missing data of self-report gout and eye diseases, the estimated glomerular filtration rate, which is a marker to help to diagnose chronic kidney disease, were lower than 6 milliliter per minute per 1.73 square meters. Incomplete physical examination data. Therefore, a total of 16,925 patients with hyperuricemia or gout and 87,194 controls were identified, shown in Figure 1. After age and sex matching, the study groups were divided into 4 groups. Health group, cases with hyperuricemia but no gout, cases with gout but normal uric acid, and cases with both diseases. We then selected the SNPs that related with gout or hyperuricemia in several genes. The proportion of SNP variation in patients with eye disease were compared with that in the control group by GWAS. The odds ratio was calculated and estimated the significance for selected SNPs. As a result, there were almost 77% males in patients with hyperuricemia or gout, and the average age was 49.46 years old. The cases with hyperuricemia or gout had more proportion of alcohol drinking, smoking, family history of gout, comorbidities, and obesity. For eye disease, cataract, dry eye, and mild dysopsia has a significantly higher prevalence of eye diseases among patients with gout but normal uric acid, compared with those with normal uric acid without gout. This result has been adjusted for age, gender, level of education, marital status, alcohol consumption, smoking status, and level of BMI in Table 2. As for hyperuricemia-related SNP variants, we found that 2 of them RS37411414 TT and CC, and RS117977 AA versus GG were associated in patients with gout but normal uric acid. And our conclusion is the results of this study indicated that gout with low level of serum uric acid is a more critical factor in ocular diseases, suggesting a potential subclinical effect exerted by its association activities in the eye. The two identified SNP variants might have association with ocular diseases and need further analysis for specific ones, such as cataract or age-related macular degeneration. Also, investigation for the underlying mechanism is needed, which may unveil new potential risk factors in the future. Okay, this is the end of my presentation, and please feel free to ask any question in detail. I'm glad to answer it. Goodbye.